The chocolate variety has steadily made its way into a few breeds over the past 20 years or so since the discovery of the gene responsible in 1996. These breeds include wyandottes, Polish, silkies, orpingtons, seramas, and more. However, one of the big problems with chocolate is that there are actually two different ways you can make a chocolate-colored chicken. One method, which is responsible for white-crested chocolate Polish, is the dun gene, which works by incomplete dominance. The chocolate found in orpingtons and wyandottes, on the other hand, is created with the sex-linked recessive chocolate gene. And both the sex-linked chocolate gene and the dun gene can be found in silkies. Both genes that cause the chocolate variety can look fairly similar, but there are some giveaways based on the way they are inherited. Because the dun gene works by incomplete dominance, when you breed two dun birds together, you should get black, dun, and khaki, which is homozygous dun. When you breed two sex-linked chocolate birds together, all the offspring should be sex-linked chocolates. Unfortunately, the rest of the genetics for sex-linked chocolate isn't so simple because the sex-linked chocolate gene is sex-linked recessive. Because the name dun isn't very appealing, in the standard, the name chocolate is used for both dun-based chocolate and sex-linked chocolate. To combat this, in Europe, the name bronze was given to refer to sex-linked chocolate. However, in America, this term as of yet isn't very well known and usually leads to more confusion. Typically, among American breeders, chocolate caused by the dun gene is referred to as dun-based chocolate, and chocolate caused by the sex-linked recessive chocolate gene is called sex-linked chocolate, sometimes abbreviated SL chocolate. For this video, we will be discussing the genetics of sex-linked chocolate, as is found in chocolate wyandottes and orpingtons, and we'll save the genetics of chocolate caused by the dun gene, like what is found in Polish, for a later date. We will also be going over a brief explanation of sex linkage, and we'll be showing 6 different punnet squares related to the breeding of the sex-linked recessive chocolate gene. To sum up what was stated in a previous video I made on the genetics of sex linkage, a sex-linked gene is a gene that is found on a sex chromosome. Chickens, just like humans, each have 2 sex chromosomes. In humans, males have an X and a Y chromosome, and females have 2 X chromosomes. Chickens, on the other hand, are reverse, with the female having a Z and a W chromosome, and a male having 2 Z chromosomes. This means that in chickens, the female's chromosomes, or genes, determine the gender of the offspring. The chocolate gene, denoted by the gene name CHOC, spelled all lowercase, is a sex-linked gene. This means that it is located on the Z chromosome. It is also a recessive gene, meaning it can be more difficult to work with when trying to introduce it into a new breed or cross with another color. The chocolate gene is also what we call a eumelanin diluter, meaning that the chocolate gene only or mainly affects the black areas of a bird. Remember, the only 2 colors on a chicken are black and red. Every color you see otherwise is either caused by a diluter, enhancer, pattern gene, or restrictor to change the color or pattern. The chocolate gene is also sensitive to ultraviolet light. You may notice birds that have been exposed to high volumes of sunlight over a period of time having a bleached appearance to their feathers. Ultraviolet light can also cause a black bird to appear chocolate, which is just another reason why keeping good records is so important. Going back to the chromosomes, males have 2 Z chromosomes, meaning they can have 2 copies of the gene or allele for chocolate. Because the chocolate gene is recessive, males must have 2 copies of the chocolate gene for it to be expressed. If a male is heterozygous for the chocolate gene, he will be black, or whatever other genes are lying underneath. The gene symbol for the absence of the chocolate gene is CHOC, spelled with a capital C and a plus sign afterwards to represent that it is the wild type. Females, on the other hand, have 1 Z chromosome and 1 W chromosome. Because they only have 1 Z chromosome, they only have 1 locus for chocolate. This means that essentially a hen can only have 1 copy of the chocolate gene or the absence of it. Because of this, we would call her hemizygous for the chocolate gene or hemizygous for the absence of the chocolate gene, depending on which allele is present. Although trying to work with sex-linked recessive genes can be tricky, the good news about the fact that the sex-linked chocolate gene is recessive is that when you breed 2 chocolates together, they will always be chocolate. If you ever breed 2 chocolates together and don't get chocolate, that means that you were probably working with dun-based chocolate instead. Although sex-linked dominant genes are usually easier to work with when forming a sex-linked pairing, such as the barred gene, sex-linked pairings can be created with sex-linked recessive genes like chocolate too. This is especially of use in breeds like silkies, where it takes so long to determine if chicks are males or females. This allows you to separate the chicks earlier in life, way before any males start thinking about fighting over females. So with that, let's go over to 6 different punnet squares related to the breeding of sex-linked chocolate. The first pair is a homozygous black male bred to a hemizygous black female. Now, because the chocolate gene is sex-linked recessive, a black male could be carrying a chocolate gene, which is why it's so important to keep good records. But in this pairing, assuming both parents are homozygous and hemizygous respectively for black or the absence of chocolate, all the chicks should be black. All the male chicks from this pair would have a genotype of C chalk/C chalk, and all the females would have the genotype of C chalk as well. This can be written out in a few ways, one of which uses a dash to fill the void for the W chromosome that does not have a chocolate locus. Some people choose to write it with a W instead to help remind readers that this is a sex-linked gene, but both are technically correct. The next pairing is a heterozygous black male split to chocolate bred to a hemizygous black female. This pairing is technically a sex-linked pairing because some of the chicks will be sex-linked, but not all. For male chicks from this pairing, 50% should be homozygous for the black gene, and 50% will be heterozygous black split to chocolate. Of course, you won't be able to tell which are which without test breeding them in the future. For the female chicks, 50% will be hemizygous for chocolate, and 50% will be hemizygous for black. So phenotypically, all the chocolate chicks are females, and the black chicks could be males or females. If you hatch a large number of chicks from this pairing and all of them come out black, that means there is a good chance that the heterozygous black split to chocolate male you are using is actually not split to chocolate and is homozygous black instead. The third pairing is a homozygous chocolate male bred to a hemizygous black female. This is the first pairing we've encountered in this video that will actually give you entirely sex-linked chicks. In this pairing, 100% of the female chicks will be chocolate. An easy way to think of this is that because a female chick gets the Z chromosome from her father, the father's alleles at the chocolate locus determine what the daughter's alleles at that locus will be. Because the father is homozygous for the chocolate allele, all of his daughters will be hemizygous for chocolate. 100% of the male offspring from this pairing will be heterozygous black split to chocolate. Because male chickens have 2 Z chromosomes, they get 1 Z chromosome from their mom and 1 Z chromosome from their dad. Because their mom is black, they get 1 Z chromosome with black from her, and because their dad is chocolate, they get 1 Z chromosome with chocolate from him. So, to sum up this pairing, all chocolate chicks are females and all black chicks are males. The fourth pairing is a homozygous black rooster bred to a hemizygous chocolate hen. Unfortunately, this pairing does not result in any chocolate chicks. From this pairing, all of the males are heterozygous black split to chocolate, and all of the females are hemizygous black. The fifth pairing is a heterozygous black split to chocolate rooster bred to a hemizygous chocolate hen. This pairing will give you both black and chocolate chicks, but none of them will be sex-linked. In this pairing, 50% of male chicks will be heterozygous black split to chocolate, and the other 50% of male chicks will be homozygous chocolates. 50% of female chicks will be hemizygous for chocolate, and 50% of female chicks will be hemizygous for black. So essentially, chocolate chicks could be males or females. Of the black chicks, all that are male are black split to chocolate, and all the females are hemizygous black. And finally, the sixth pairing is a homozygous chocolate male bred to a hemizygous chocolate female. And as stated earlier, all chicks, both males and females from this pairing, will be chocolate, with the males being homozygous chocolate and the females being hemizygous chocolate. Overall, the sex-linked chocolate gene found in wyandottes, orpingtons, silkies, and more is steadily increasing in popularity, not only for its beautiful brown color, but also for the fact that it can be used to create sex-linked pairings, which is so useful in breeds like silkies. Because chocolate only affects eumelanin, many different varieties can be made using it. Although most of the development of different colors using the chocolate gene have been done with seramas, we have also seen the chocolate paint variety in silkies, as well as the mauve variety, which is created using sex-linked chocolate and one copy of the blue gene. Although the chocolate variety caused by sex-linked chocolate is not yet recognized in a lot of breeds, it seems like one that may be worked on in the near future. As always, thank you so much for watching, and I hope to see you in the next video.